Okay, so we are now recording, and thank you very much, Federico, for agreeing to give a Belluso seminar, and you can get started.
Okay, thank you very much, Kate, for this very nice invitation. I'm very happy to present you my results. So, yeah, as we were saying, I was asked to give you a little bit of my background before starting the actual topic of my talk. Well, first, I would like to thank the co-workers that took part with me in this project about self-division of giant vesicles. And especially Ilenia Miele, that she used to be my PhD student, now is a postdoc in Salerno University in south of Italy. And then this work was done in collaboration with the group of Professor István Lagsi from Budapest University of Technology and Gabor Hollo, who is a PhD student in the group of István. So my background, presently I am an associate professor in Siena University, which is, maybe you know, Siena is a very nice town, small little town in the center of Italy, but full of history and many very nice things to see and to visit. And we produce a very good wine and food over here. So if you're interested in visiting Italy, you should come in Siena for sure. But my research activity mainly is concerned with far from equilibrium chemical processes. And I started since my master thesis to work on oscillating chemical reactions, the Belluso-Zappotisky reaction in particular. When we talk about far from equilibrium condition in chemical systems, we mainly think about CSTR reactors that are reactors continuously supply with reactants that keep the system far from equilibrium. And in this condition, you can have very, very nice and exotic manifestation of nonlinear dynamics and very nice, let's say, behavior of chemical systems and quite complex. For example, you can get oscillating chemical reaction. This is an example of the Belluso-Zappotisky reaction that change color in time due to the change of the redox state of some of the intermediates of the reaction. And when you couple this nice oscillating behavior with diffusion and transport properties, you can get what is called pattern formation. Pattern formation is the born of a chemical structure that can be either moving or stationary. In that case, we talk about a Turing structure in simple solutions, very simple. And so you can get these very nice patterns forming like spirals, target patterns, and chemical wave in general. Basically, to have oscillations and pattern formation, you should have a system with nonlinear kinetics, for example, autocatalytic systems, where there is a mechanism at play done by the alternation of activating and inhibiting processes. So certain species, certain chemical intermediates act as activators. For example, they promote autocatalysis, and other species act as inhibitors, and they depress the concentration of the autocatalytic species, just an example. You can get several chemical oscillators. Now there is a complete taxonomy of this system, of these systems. You can have redox oscillating reaction, pH oscillating reaction, precipitation oscillating reaction, and so forth. And why we use this kind of system? Well, for sure, because they are very nice to see and to work with. Probably you saw many of these movies in educational presentations for students, but actually even in the research activity, they are very, very used as a model system for complex behavior in chemistry. Now, this discipline was very big in the 70s and in the 80s after the Nobel Prize winning Ilya Prigozhin somehow gave the basis, the theoretical basis to understand such phenomena. And he got the Nobel Prize in 77, sorry, for the concept of dissipative structures. But now there is a full reprise of these systems because they are used, for example, in the application in chemically fueled self-assembly for nanoparticles, for example, in the search for autonomous systems that can perform complex tasks far from equilibrium, with many applications in medical fields, material science, and so on. And in general, they are a very good model for self-organization in chemical and biochemical systems. So I told you that CSTR are the most used system for studying processes far from equilibrium in chemistry, but actually we have another system that we are exploiting, and I will talk a little bit better about this later on in the presentation, that are the semi-batch reactors, where you have selective permeabilities of membranes. I mean, you use membranes to compartmentalize your chemical reaction, your reactor, and you use the permeability of such membranes to allow selected species to cross the barriers and feed the system, and this can be both ingoing or outgoing chemical species. So, as I was saying, I'm using such autocatalytic inorganic reactions for many applications. I started to study chemical chaos with these reactions, but now I moved to study models for biological systems. I mean, I use such inorganic reactions, which are much simpler than biochemical systems, to make proof of concepts of some functions and behavior of more complex biological entities, like for example, cells. And a few years ago, we started to use this Belluso-Zappotisky reaction. Now there is a sketch of the chemical mechanism. I will not give you details, boring details about that. It's just to show that there is a cycle between three different states where you have activation and inhibition that basically allows for the formation of oscillating behavior. And in the Belluso-Zappotisky reaction, you have the shift between red color and blue color depending on the redox state of the catalyst, which is red when it's reduced, this is ferro in catalyst, and blue, which is oxidase. So in this way, you basically have a system that is able to generate signals, because you have an on-off process that can release different intermediates in different stages of the reaction. So basically, it is a signal generator, but it is also an antenna for signals coming from outside. So this system is able to react to external stimuli that can be inhibiting or activating stimuli. So we took this reaction and we put inside of lipid giant vesicles made, I don't remember, in this case, they were made by DMPC phospholipids. And then we allow single entities to communicate among them. That was a proof of concept for studying chemical communication in a network of micro oscillators. The property that we use of the Belluso-Zappotisky, as I was saying, is the generation of intermediates like bromous acid or some radicals to spread signals in the environment. They can be spread in a homogeneous way or in a targeted way, for example, using waves to spread targets on a straight line. And these messengers can be of inhibitory nature or activatory nature to generate, for example, antiphase oscillation in Turing structure when the inhibition is at play, or in-phase oscillation and signal amplification. This is, for example, this was used, for example, to explain some features of the neural communication when to understand the functioning of the brain of mammals and human beings in general. And for example, what we did was to studying arrays or a population of different giant unilamellar vesicles coupled together through the passage of intermediates along through the membrane. We explored arrays, so one-dimensional system, or colonies, 2 or 3-dimensional systems. And what we did was to study the oscillation into each single vesicle, and then make a phase analysis of the oscillation to understand what was going on at the global level. And the second step that we recently developed is to understand how the membrane properties, properties at the nanoscale of the system, can impact the global behavior of all the system. And so we prepared different vesicles having different dopants in the membrane, which change the conformation of the membrane. That can be multilamellar, monolamellar, and so forth. We can have charged surfaces or completely non-polar surfaces. And these, of course, change the coupling strength among systems. And then we studied the behavior of, in this case, 4 different oscillators coupled together to see how they behave when you have external stimuli. And this is just a resume that show what we recently got in our results. But this was just so you can have, for example, in-phase oscillation with clustering resonance, or a complete inhibition of the system where you have antiphase oscillation. This is the way that is thought that brain works. So communication among neurons follow an inhibitory coupling due to electrical signals. So this is more or less my background, and I worked with this system until a few years ago. Now, still in the light of making simple models for complex systems, we shifted towards more biological systems, and we were interested in understanding the process of cell division and maybe replication, but still using a very simple chemical system, so not biological system. So now I'm going into details of the topic of today, which is the division of the self-division of giant vesicles accomplished by simple internal chemical stimulus. This is the concept of our work. So we put a chemical system inside. Our idea was to put a chemical system inside a giant vesicle, and through the passive diffusion of some precursor inside the cell, we wanted to trigger a reaction that changed the pH in this case, and see if this pH change could induce some instability in the membrane to bring to cellular division. So there are at play several let's say several themes, several topics that I explained you before. We are in the presence of a nonlinear mechanism. We are using a stimuli responsive membrane, and of course this is a far from equilibrium system, kept far from equilibrium through passive diffusion. And then we used this giant vesicle in the range of 10 to 40 micrometers in diameter. So to accomplish this task, we follow with 3 steps. First, we choose a chemical reaction. In this case, is an enzymatic reaction. So it's not as simple as a pure chemical reaction, but still we could master the kinetics of this system, because this reaction is very well known and very studied. I'm talking about the urea-urease reaction, that basically is the hydrolysis of urea, this molecule that takes place in the presence of the enzyme urease. And this hydrolysis release ammonia and CO2. So the net result of this reaction is to produce molecules able to increase the pH of the surrounding solution. Now, an interesting feature of this reaction is that when you the kinetics of this reaction is ruled by a Michaelis-Menten type reaction rate, where you have, of course, the classic Michaelis-Menten implemented with some terms that account for inhibition and pH dependence, but it's not important now, those details. What is important is that the reaction rate depends on the pH. So this is the typical bell-shaped curve that describes the reaction rate as a function of pH. And when you work in unbuffered media, you can start, for example, you can set your initial condition of the experiment in acidic condition, and when the reaction takes place, let's say we climb this branch of the bell-shaped curve, and so the reaction rate increase and the pH increase as well, until you get in a point that you get a real autocatalysis. When you are around this area, the reaction rate is maximum, and so you have a sharp increase of the reaction, followed by a plateau, because you go into the right branch of this bell-shaped curve, and so the reaction slow down. Here I signed the clock time, which is a jargon term to describe the lag time between the beginning of the reaction and the point, the inflection point of the curve, which is the point where the reaction has the maximum rate. So it corresponds to this point more or less in this bell-shaped curve. Now, this kind of reaction, of course, are very interesting, because they are tunable and programmable according to the initial condition that you set in your experiment. So, for example, they depend on the pH, and the length of this clock time follows the initial condition. So it can be shorter or longer, or faster or slower, and so on. So let's say it's a tunable system. The second step was to find a proper stimuli responsive membrane. Our choice went in the direction of using fatty acids, in particular oleic acid, because it's well known that fatty acids, the chemical behavior of the fatty acids depends on the pH of the solution. In particular, the self-assembly behavior of such a molecule is strongly dependent on the pH. And you can have a phase separation when you are below pH 8, for example. At intermediate pH, you have that half of the fatty acid is deprotonated and half is protonated. So you can build the system build this nice hydrogen bonding that gives rise to vesicles, actually. And when the pH is higher, you can get micelles of pure oleate molecules. So the feature that is interesting for us is that they respond to pH stimuli. Moreover, we decided to use a blend of this fatty acid with with phospholipids, POPC in this case, for several reasons. Because first of all, because they are more stable, such membrane, and then they are considered as as a good prototype for protocellular membranes. So since our work goes in the direction to find a simple mechanism, as I told you, to explain a very complex behavior, this can have a relevance for origin of life studies, so for all the abiogenetic theory. And of course, in this case, such a system can be very, very interesting. So they are stable, but yet they allow some degree of permeability towards external molecules. The third step was to be sure that such clock behavior, such autocatalysis can take place inside a confined environment. And so we tested if permeability and enzymatic reaction still worked when confined in giant vesicles. This is an example of a population of pure POPC vesicles that starts from acidic condition. When you see dark image, it means that the pH is in acidic condition. When you see the intensity of the fluorescence increase in time, it means that the pH is increasing. So our system worked pretty well, and we could prove the permeability of, for example, acetic acid, a weak acid that we use to set initial conditions. And above all, we proved that urea, that substrate for the enzymatic reaction, was actually able to cross this membrane. Urease, the enzyme, was firmly confined into the membrane, and we proved with several experiments that urease is not able to cross membrane. So the situation is more or less this one. You have the enzyme with the fluorescent probe encapsulated into the giant vesicle, and you have a species that can cross membrane. And as you can see, the clock time is a function of the initial acid concentration. So you have very fast reaction when the acetic acid is low, and you have very slow to known reaction when the concentration of acetic acid is too high. So now we are ready to start experiments with mixed membranes, and to produce our vesicles, we used a phase transfer method that Kate probably knows very well, because was one of the most used system in a group of Pierluigi. And of course, if you want details on this method, we can discuss it later. What is interesting for us is that with this method, you can produce an heterogeneous population of vesicles, and you have a distribution of the diameter of the vesicles according to a Poisson distribution. And in our experimental condition, we worked mainly with vesicles having a diameter around 10 to 15 micrometers, but also smaller and larger vesicles. And then we calibrated the pH probe that was pyranine inside the vesicle to establish a relation between pH and the fluorescent intensity to have a quantitative account of our experiment. In a typical experiment, we put a solution containing a population of vesicles like this into a microscope slide. We add urea to initiate the reaction, and then we observe experiments at the confocal or optical microscope. This is the outcome of a typical experiment. And you see that after the pH starts to increase or the reaction takes place, these vesicles undergoes shape transformation, and they start from a spherical shape, then pass to a prolate shape, and then to a dumbbell shape. We couldn't get let's say a definitive division because still you need to add some energy to the system to break the neck that forms between the two daughter vesicles. And for example, we accomplished this with a laser beam. So it's enough that you irradiate one of the two vesicles with the laser beam and they detach and move apart. And here is some statistics that is not very important, but we always got 25% of success independently, irrespective of the size of the vesicles. And there are optimal experimental conditions that allow to obtain successful divisions. For example, the summation of the concentration of POPC and oleic acid in the membrane and the ratio between them. I will say a little bit more about these in a later slide, but as you can see, we have an optimal composition to get divisions. Another step was to check if the division was actually due to a pH change. So we recorded the fluorescence image, and you can see we start from dark level of fluorescence, and as the reaction progresses, we go towards basic pH. And what we got was a difference of from 0.5 to 1 unit of pH, which is not bad. And most important, what we could find is that is enough, 1 unit pH is enough to deprotonate 20% of the oleic acid that was present in the membrane. And in our plan, this deprotonation was at the basis of the instability that causes such division. Moreover, we can see that material is retained during the division because both the daughter vesicles continue to be fluorescent, and so it means that the reaction still goes on, and enzyme went into the passed from the mother to the daughter vesicles. Okay, the mechanism. I will try to explain a little bit more in detail the mechanism that we think is at play. We start at pH 6, more or less, and so the membrane is composed mainly by the POPC phospholipid and oleic acid in its protonated form. When the reaction takes place and pH starts to increase, for example, at pH 6.5, the oleic acid, which is in the inner leaflet of the membrane and is in contact with the water solution inside the vesicle, in the lumen of the vesicles, they start to deprotonate, and their solubility in water increase. So somehow they detach from the membrane and goes into the lumen of the vesicle, and this will cause the instability. So you have some molecule of oleate that move from the inner layer of the membrane and goes into the lumen, and this causes that some molecule of the oleate in the outer leaflet of the membrane replace these vesicles to keep the membrane stable. At one point, you can also have the formation of aggregates, of oleate aggregates in solution inside that somehow accelerate the solubilization ratio of the oleate molecules. Another important point is that the deprotonation of the oleic acid cause an increase of the average area per molecule. So since there is a repulsion between negatively charged oleate molecules, those tend to repel each other, and so the inner area increase a little bit. Now, this is not enough to cause the division, the complete destabilization of the membrane followed by division. So according to this theory, ADE theory, which means area difference elasticity theory, in general, the equilibrium shape of a vesicle equilibrium shape, I mean spherical, prolate, dumbbell, divided, and so on, can be described by two parameters, which are the volume of the vesicle, which can be controlled by, for example, by osmosis, and the difference in the preferred area difference of the two leaflets of the membrane. So the membrane, as we were saying before, is composed by two amphiphilic leaflets that somehow are in equilibrium among each other when the shape is in equilibrium. If you cause a change in the composition of one of the two leaflets, somehow you introduce a tension between the two leaflets that want to rearrange in a new shape, in a new global shape of the vesicle. And this is the parameter we affect with the pH change. So the pH change causes the instability in the inner leaflet, and this in turn provokes a tension between the two leaflets that wants to rearrange each other to obtain an equilibrium structure. And you can draw a phase diagram where you have the difference in the area of the leaflet and the volume to see which kind of form is preserved, is maintained in specific condition. And in particular, if you start from a perfect sphere, you need to decrease the volume of the vesicle and to increase the area difference to obtain the dumb shell shape. So with our pH change, we somehow increase the difference in the two leaflets, and with osmosis, we decrease, by controlling osmosis, we decrease the volume. Why we impact the osmotic pressure in the vesicles? Because when we add urea, even though urea is permeable to the membrane, however, its permeability is much lower than the permeability of water. So at the beginning of the experiment, you have a disequilibrium between the osmotic pressure inside and outside the system that eventually will be equilibrated when urea goes inside. But in the first stage of the experiment, you have that water molecule exit from the vesicle to compensate the osmotic pressure, and so you have a deflation of the vesicles. So your volume somehow, your vesicle shrinks and the volume decreases. We performed several control experiments. Even here, I will not go into details, but just to point out that in our experiments, osmotic pressure and chemical stimulus should act in synergy. They should be at the same time, they should take place at the same time to have a synergic effect and to actually bring the system to divide. So say that we tried some modeling of our system, and we put together all the things I told you up to now. So here we have the chemical kinetics inside the vesicles, which accounts for the enzymatic reaction, and the net result is to increase the pH. Of course, we consider also the transport processes of the chemical species from outside to inside and vice versa. And here we draw a set of differential equations to account for the movement of molecules from the outside leaflet to inside leaflet, and from here to the lumen of the vesicles. So this differential equation accounts for the movement, the flip-flop of the molecule in the leaflet, and the solubilization of the olate inside the molecule. And then we have a function that describes the increase of the inner area leaflet, and we got parameters through Wilhelmy thin balance and molecular dynamics simulation. So here we have the results of our simulations, and to obtain the equilibrium shape drawn in this movie, we minimize the Elfrich energy that accounts for the shape of the vesicles extracted from this theory, from the ADE theory. And what you see is that at different volume and area difference, you can get a different shape, spherical, prolate, this kind of nutshell, pear shape, until you get the complete division. And this animation I will show you again how the actual experiment on the left is well described by this the minimization of the Elfrich energy in time. And here you have pH, osmotic, the difference in the osmotic pressure inside and outside the vesicle, and this is the behavior of the difference in the area leaflets. By using this model, we introduce, and I'm going to conclude the presentation. We introduce another factor which accounts for the bending modulus of the membrane. Of course, the capability of the membrane to somehow, well, let's say the instability driven by the difference in the area of the two leaflets depends on the elasticity properties of the molecules that compose the membrane. So when we change the composition of the membrane, actually we change this parameter in the Elfrich energy, which accounts for the bending modulus of the whole membrane. So we also experimentally found a relation which relate this modulus with the composition of the membrane. And through some simulations, we extrapolated all the values of the maximum of the area difference, of the preferred area difference, and we could obtain such a phase diagram, which explain why we found an optimal composition for the division success rate that I showed in one of the previous slides. So there is a narrow range of parameters where you can get nice division, and you cannot get outside this parameter basically for two reasons. Because when this composition is shifted towards a lot of POPC, you don't have enough oleic acid that get protonated, and so the maximum area difference is too low to guarantee the division according to the other ADE theory. On the other hand, when you have too much oleic acid, the membrane is much more elastic, and so this causes that the membrane is less prone to get divided. So this explains why you can get this behavior. So our outcomes is quite interesting from let's say a basic research point of view, because point out that you can get a successful division, for example, in a prebiotic environment where you don't get, of course, genetic material or a complex enzymatic network that actually drive division and replication in modern biological system. But you need, you can still have a process of division driven by simple chemical mechanism and osmotic processes, which could be very common in prebiotic condition, provided that the membrane the membrane properties in terms of bending and elasticity fall into a specific range. So just to draw some conclusion, as I told you, we could get effect in division by an internal stimulus. I stress this word internal because Many of the papers that were published, most of the papers that were published in the past years could obtain an effective division of micelles or vesicles, but always using external physical stimuli. For example, temperature change or addition of precursor of some membrane molecule and so forth. This is one of the first examples where the division is triggered by an internal stimulus, an internal chemical mechanism. Osmosis and chemistry, they have to play together. Even in this case, there was quite a long debate in the past if osmosis or other mechanisms could drive division effectively. And in our opinion, you should have both, because if you don't have attention in the leaflet, it's difficult to get a division. And of course, these open up, our findings can open up the way for new kind of experiments. We can exploit the tunability of our system to make a programmable system that can open and divide only after a certain time. This might be important in drug delivery processes, for example. And of course, you can have plenty of autocatalytic reactions to use with the same approach or different membrane composition. So I thank you all for your attention, and of course I'm open to questions and curiosity and so forth.
Thank you very much. This is a pretty cool system, and I have a lot of questions myself, but I would like to let other people go first. There is several questions in chat. Can you see chat?
No. I will stop sharing the screen. Yeah. So let me stop share and should be able to. Okay. Okay, I see the chat.
If you could read a question out loud for the recording, that would be great.
Okay, the first thanks for this is a fascinating work. Thank you for presenting. Thank you very much for this compliment. In the division experiments, does the lumen remain intact or does content leak some? So in one of the slides, I showed you that the content remain intact. However, we experience some systems, especially when the concentration of oleic acid is quite high, where we have bursting of the vesicle. So actually, some of the content go spread around. We couldn't quantify if the transfer of the enzyme is quantitative from the mother to the daughter vesicles because we didn't know how to measure it. But for example, we could quantify the fluorescence of the vesicles before and after the division, and it was quantitative. So at least almost quantitative. So it means that at least pyranine, that is the fluorescent probe, is transferred from the mother to the daughter vesicles. I hope I answered this question. I go to the second. Could the pH change be induced with an ion channel? I think yes. The answer is yes. There is a very interesting work from other people from Italy, from an Italian group. One is Pasquale Stano and Fabio Mavelli and Emiliano Altamura. I guess Emiliano Altamura is going to give a seminar in one of your next meetings. In this system, they managed to, I don't want to spoil his presentation, but I can say this, that they managed to embed a chromatophore, a bacterial chromatophore system in the membrane of the system. And so this system is able to convert visible light into a pH gradient. So we have already started a collaboration with them to try to mix together our system. So my answer is yes, I think it's possible, because you can get a proton gradient inside the vesicle with this system, and so I believe you can do that also with an ion channel. It would be very interesting to test this. Great results and presentation. Thank you very much. Residual lipid nanotubes are among the challenges of attained division on cell mimics. How can the formation of lipid tube nanotube connecting daughter vesicles be ruled out in your pH-induced division assay? If I get your question, you want to know if there is if the neck that connect the two daughter vesicles is still open or is closed. Am I correct? Let me see if there is an answer in chat. Okay. Okay, so this was actually a question that a referee that reviewed our first paper on this work asked, and was and it's a very good question. So we proved that the two daughter vesicles are not in contact with photobleaching experiments. So at the confocal microscope, we waited for the full division, and then we isolated two daughter vesicles connected together, and we photobleached one of the two and waited for the recovery of the fluorescence, but we didn't see any recovery. So it means that the pyranine from one of the from the other vesicles cannot cross the membrane. So the answer to this question is yes, they are not connected, and material is retained. And this is also the experiment I was mentioning before, when I say that if you irradiate with the laser beam the two vesicles, they move apart from each other. So this was also the proof that if you give an extra energy to the system, you can get actual division. This was, for example, there was a very nice work from the Shostak group where they could get purling of the of micelles, all of them connected, and they provided extra energy by shaking the system. I don't know if you are aware of this system, but the point is that you generally need an extra source of energy to have the complete fission of the neck. But in our case, they were not connected. Would it be possible to use different fatty acids instead of oleic acid? And if yes, how this would impact division of vesicles? Thank you for the great presentation. Thank you for your question. I guess yes. We didn't try actually. I think the impact of different oleic acids is would be the working pH. I mean, the pK of the fatty acids depends on the hydrocarbon chain that they have, and so the longest the hydrocarbon chain, the lowest is the pK, and vice versa. And so I guess that if you use different fatty acids, you need to find a proper autocatalytic pH reaction that works in another pH range. That's my guess. And of course, you need to find fatty acids that can be embedded in the vesicle membranes. So you can also change, in this case, the phospholipid that you want to use. We worked with this system because it's well known that POPC and oleic acid can form blended membranes, as in the paper of Shostak that I showed at the beginning of the presentation. Harold Muriel, this is a really good model for prebiotic division. Fascinating. Thank you very much. Does osmotic pressure affect the division? For example, if vesicle or outside is stuffed with high osmolarity solution like sucrose. I think I answered this question during the presentation, but I say it again. Yes, osmotic pressure is fundamental to get division. And actually, one of the control experiments that I showed, sorry, I didn't go into details, just to be not too much boring, but it was to buffer the osmolarity of the solution both inside and outside of the system, and we couldn't observe any division. In the absence of osmotic pressure difference, what we observe is some deformation of the vesicles, but we couldn't get any successful budding, let's say, or division, if you prefer. So yes, osmotic pressure is fundamental. Chris, does addition of sterols affect divisions? Increasing POPC membranes fluidity with cholesterol with the lower fatty acid requirements. This is also a very nice experiment to try. We didn't, but I can say that yes, cholesterol is known to increase the stiffness of the membrane and the rigidity, so it might be interesting to try. Actually, I worked with cholesterol in previous experiments about communication in coupled oscillators, and the effect of cholesterol was to damp communication between the two systems, but it has nothing to do with divisions, of course. But probably yes, increasing the rigidity of the membrane will allow to use less fatty acid. But as I said, we still need a certain amount of oleic acid, because otherwise the membrane is not enough pH sensitive. I mean, the number of oleate molecules that form and pass to the solution is too low to guarantee the necessary tension between the two leaflets. So probably yes, but with some cautions. Really good work. Thank you for presenting. Thank you very much, Ivan, for your comment. Without applying laser energy, does the final membrane scission take place in these membrane vesicles? If not, then what other accessory component is necessary for the division? Yeah, even to this question, probably I answered in one of my previous comments. We need an extra energy to do that. We need to go beyond the Boltzmann threshold for obtaining division. This is common even in real, in modern cell division processes. In that case, you have, of course, you have specific enzymes that promote these, but still they need to be fed with some ATP energy and so on. That's because at the end, the budded vesicle, so when you have the neck connected, it's an equilibrium structure. So you need to go farther from equilibrium somehow. When the pH reaction ends and urea is consumed, of course, you go to the chemical equilibrium. And as I told you, other people use the temperature to do that. Increasing temperature I mean, the system was a little bit more complex, but basically increasing temperature, you get division because of the different state of the lipids. You pass from gel state to liquid state, and so this instability causes the division. Others like Shostak shaked the specimen with the samples inside to provide the mechanical energy and so forth. So those are the most used system to give this extra energy. There is a work, there are many works from a Japanese group Sukavara, I think is his name, the PI of the group, that they actually used a very complex system that they synthesize ball-amphiphilic molecules directly into the membrane. It's a very nice, very nice work. I guess it was published in Nature Chemistry, the first experiment. And so they provide this extra energy by feeding the system with the precursor of this ball amphiphile. So it's another way to move the system far from equilibrium, feeding from external dissolution. So for the revision, so Brian is asking, fascinating work, thank you very much. Once the vesicles divide into dumbbells, can you change the external environment so that they could reform into a giant vesicle? In some cases, we don't even need to change the external environment, because some of the divided vesicles go back to the initial state spontaneously. That's probably because the composition of the membrane, of course, we have to say that the composition of the membrane is not homogeneous in the population. As I was telling you that the method we use, this droplet transfer method that we use to produce the colony of the vesicle, is actually very powerful, very easy to use, but has some drawbacks. One is the polydispersity in the size of the vesicle, and the other one is that you get inhomogeneous distribution of the reactants and of the precursors. So I guess you can work even from external environment. For example, if you add more POPC or fatty acid, you can change the composition of the membrane. You can somehow reform the divided vesicles. And my guess is just because we see a spontaneous, such kind of spontaneous transition even in normal experiments. So I guess yes, the answer is yes, yes, and probably you can act with these, and probably you can also act with some pH changing molecule. You can buffer the solution, see what happens if you send the system back to initial pH. That can be interesting to explore. You mentioned drug delivery. Is fatty acid membrane stable enough for bloodstream? This would be really interesting, especially for things like tumor microenvironment delivery, where pH is significantly different. Really interesting application. Yes, indeed, is one of the applications we had in mind, or that we use to try to sell our work, even for practical application. I'm not sure fatty acids can be stable in blood and in physiological fluids, because they are very sensitive to divalent ions. It's known, it's well known that fatty acids precipitate in the presence, for example, of magnesium and calcium. So they are very sensitive to divalent ions. However, the work of Shostak that I showed you at the beginning of the presentation, it's specifically about that. They found that blended membrane, POPC, and oleic acid, oleic acid and the PPC membrane, blended membrane, are more stable in the presence of divalent cations. So this may let's say open up some road to get this system even more stable. There is another important point to address. We cannot use giant vesicle for drug delivery applications, because we know that they are too large, and in the immunological system of human beings immediately attack them and destroy them. So at present, our main concern is about demonstrating that this division takes place also in nanovesicles. So as a proof of concept, we use giant vesicles, but we want to study nanovesicles because they will be eventually used for real drug delivery application. And as you said, for tumoral treatment, it would be very effective because it's known that tumoral cells have a very different pH with respect to the physiological environment. So I guess I answered to all the questions. Of course, if you want to have to make further questions or things that I just said, they are very welcome. Thank you for these very, very interesting questions.
Thank you very much. I have one quick question. Did you ever try to do a pH-driven division after growing POPC vesicles with fatty acids? So the kind of a fusion of this growth system. And then once you have enough fatty acids, could you then divide them? Or do you see some obvious reason why this has no chance of working?
No, I think we didn't try. Thank you very much for this question, which is actually a very nice suggestion. And we didn't even think to that. But so that would be, you mean that this could be a very nice mechanism where you use both, where you have both growth and division together?
Yes.
That would be very cool. That would be very cool. We didn't try, but I think it can be interesting to try this. Yes, thank you very much for this suggestion more than question. For your comment.
Thank you, and I'm excited to see more on that system.
Okay, hopefully.
Thank you very much, everyone, and have a great Monday. And thank you very much again, Federico, for the fascinating talk.
Thank you, Kate, and thank you for your invitation. Thank
Bye,
you very
everyone.
much. See you soon. Bye.
Bye.
Bye.